Welcome to this biochemistry video on oxidative phosphorylation. This topic is quite in depth, and we're going to probably fly over it a little bit higher level anyway than what we do on some of the other topics. So to begin with, we just want to put things in perspective on where we are. The oxidative phosphorylation, also called electron transport chain, is right at the end, and this is where most of the ATP is produced as we get through all of the catabolic processes. It's uh, it's the second part of what we call the common catabolic pathway, which includes the citric acid cycle and then goes into oxidative phosphorylation. The electron transport chain and the phosphorylation are usually grouped together in this. So there's numerous names, and they do have specific meanings. They're used pretty interchangeably, especially by me. I usually call it oxidative phosphorylation down through this whole process. This is a schematic diagram of the electron transport chain, and you can see there's 5 complexes in here. Usually there are 4 that are referenced, complex 1 through 4. And the 5th one, while sometimes called complex 5, is generally referred to as the FOF1 ATPase. And that's the way I talk about it. Most people, or some people, put it as complex 5. This kind of shows the different complexes and the different molecules. It kind of gives you an idea of the flow of electrons and the flow of the protons, and gives you an idea of where it sits in the membrane. Oxidative phosphorylation happens on the inner mitochondrial membrane. That's an important understanding. It happens between the intermembrane space. The mitochondria has two membranes, an outside membrane and an inner membrane, where there's a matrix, and this happens on the inner membrane, and it happens into the intermembrane space. And we have an electrochemical gradient that's created across that with the protons, and that's what's used to create the energy. So I like showing this version, this protein map, and the general idea here is that you can see that all of these things are proteins, and they're very large protein complexes, and they do a lot of work. And that's kind of the important part. It also labels all of them. I'm not going to go into that much detail, and I don't think we will in the lecture as well. But it does give you, this is important because it gives you all the enzymes that are involved and their numbers and the products. And so if you ever need it as a reference, you can look it up and see what proteins are utilized. All right, so let's talk about complex 1. Complex 1 is an NADH dehydrogenase, and on the left-hand side, this is the way we're going to present all this. On the left-hand side, you see that there's a complex here that's got 40 subunits. There's a flavoprotein. There's several iron-sulfur clusters. There's a coenzyme Q, ubiquitinone. What generally happens here is we go from NADH to NAD plus and a proton, and we produce numerous hydrogen or numerous protons. So let's just walk through that. So we have the oxidation of NADH, and that allows to move 2 protons from the membrane matrix into the intermembrane space. So complex 1 oxidizes NADH to NAD+, and you can see that happening on the right-hand side. The oxidizing agent is CoQ, which is reduced to CoQH2, and as the electrons pass through the complex, this allows 4 protons to pump into the intermembrane space. So we produce 2 protons, and we also pump protons. We're not quite sure how this works in the protons, but there's definitely conformational changes in the complex that allow the binding of the protons on the N-terminal side of the membrane on the N side, and allows them to release on the P side. So also we have electrons that are transferred from the chain of iron-sulfur clusters that we talked about, and you can see those in the right-hand diagram pretty effectively. Those allow the electrons to be transferred from those iron sulfur clusters to ubiquinone, which is the molecule. It's in the membrane. That's labeled Q and QH, and the reduction of that ubiquinone also contributes to the generation of the proton gradient, because 2 protons are taken up from the matrix as those things are reduced. So that's the general NAD hydrogenase. You see the equation on the bottom. We're basically going to create 4 protons through that thing, through complex 1. So 4 protons are pumped from the inside to from the mitochondrial matrix side to the intermembrane space. All right, complex 2. Complex 2, the enzyme we have is succinate Q oxidoreductase. It's also known as complex 2 or succinate dehydrogenase. It's a little bit unusual because it's the only enzyme that's part of the citric acid cycle and the electron transport chain. So you might recall this from the glycolysis lecture. And basically we're going to get succinate and we're going to convert it to fumarate. And we end up again with iron sulfur clusters. We end up with coenzyme Q being transferred to coenzyme QH2, and we end up with some protons generating. 4 protein subunits, and it also contains an adenine dinucleotide FAD cofactor. It does have iron sulfur clusters, and it does have a heme group. That doesn't participate in electron transfer, but it's important in decreasing the production of reactive oxygen species. So oxygen succinate to fumarate is the major output, and we do reduce ubiquinone. This reaction releases less energy than the oxidation of NADH, so complex 2 does not transport protons across the membrane and does not contribute to the proton gradient. Complex 3. This delivers electrons from CoQH2 to cytochrome C. So we're now transferring the electrons. This is Q cytochrome C oxidoreductase is the enzyme. It's catalyzed by the oxidation of 1 molecule of ubiquinol and the reduction of 2 molecules of cytochrome C. And then cytochrome C is a heme protein that's usually associated with the mitochondria. So complex 3 and 4, you can see on the left-hand side we have the ubiquinone being transferred and cycled through. You can see the different subunits of this protein. Complex 3 does transfer the cytochrome C over to the cytochrome C complex on complex 4, and that provides the next step for the reaction, which is done by cytochrome oxidase. And in the cytochrome oxidase has 13 subunits. One is a cytochrome A3, so electrons flow from cytochrome C to complex 3, and from A3 the electrons are transferred to oxygen O2. This enzyme mediates the final reaction of the electron transport chain, and it transfers electrons to oxygen while pumping the protons across the membrane. So the final electron acceptor in this process is oxygen, and this provides the last step. It's the redox reaction, and it's also where hydrogens are pumped from the matrix into the intermembrane space. And so if we sum up the reactions of the complexes 1 through 4, we get 6 hydrogen are pumped out per NADH and 4 per FADH2. And now we're off to the FOF1 ATPase. It's also known as F-type ATPases or ATP synthase. It's an ATPase that's found in the mitochondria, and it uses a proton gradient just exactly like a molecular motor to drive ATP synthase production. synthesis, and it's a passive flux of protons across the membrane down an electrochemical gradient, and therefore energy is released by the transport reaction, and that is going to be used. The conformational changes are then used to generate the ATP energy. The FO domain is the integral membrane. It's one on the top that you see. F1 is the one that sits in. It's got FOF1 particles are mainly formed by polypeptides, and the FO1 particle contains 5 types of polypeptides, basically protein subunits, and you can see them in the picture in blue. You've got your alpha and beta in different conformations. This happens to be a hexamer, and they form a rotary motor. And so and you can see the motor, as these protons go through, they cause the stalk to rotate, and this rotation is basically a camshaft for the F1 domain. And ADP and an inorganic phosphate then bind spontaneously and then undergo like a conformational change. So those are the 4 complexes in general. We'll definitely go over in greater detail in them in the lecture. The overall reaction for oxidative phosphorylation is shown below. We start with NADH. We end up with 3 and ADP, and oxygen, phosphate, hydrogen. We end up with NAD+, 3 ATP, and water. FADH, very similar. We end up with FAD and 2 ATP and water. So oxidation of each NADH gives 3, and each FADH2 gives 2. And that allows for a large number of ATP to be produced on the fly. Again, the total energy created is 12 ATP. So at the end of the day, that's for each 2-carbon acetyl unit entering the citric acid cycle. This slide has a great summary of oxidative phosphorylation and the molecules that are associated with it, the enzymes, the products, the reactants, the enzymes. I would know this chart for the exam, as well as the entry points for this process. And with that, appreciate you listening. Have a great day.